In this video, we are going to learn about the class of biological molecules called proteins. Proteins perform a multiple number of different functions in living systems. They play a role in creating structures of biological activity, structures that perform biological activities. They're involved in transport of organelles, molecules across the cell membrane or within the cell. They are part of the defensive systems of our bodies. Most enzymes are made of proteins, and enzymes are molecules that catalyze chemical reactions in our bodies. They're involved in the contractile activity of our muscles, and they also play a very important role in communication between the cells and within the cells. So they play a large number of different roles, and they are very central for the functioning of living beings. So proteins are polymers, and the monomers that are used to make proteins are amino acids. When we attach a number of different amino acids end over end using covalent bonds, we form a polypeptide, which poly means many. Polypeptides is long chain of amino acids. What do amino acids look like? Let's spend some time to go over the different parts of an amino acid. First of all, in any amino acid, there is a central carbon, which is called the alpha carbon. To this carbon, there are a number of functional groups that are attached, which is common in all amino acids. In nature, there are 20 different amino acids, and among all the different amino acids in nature, these three parts of an amino acid are exactly the same. First is an amino functional group. Then there is a carboxyl functional group, and that's the reason we call this an amino acid. The carboxyl functional group is acidic, amino group is basic. And then there is a hydrogen that is attached to carbon. So these three parts are exactly the same in all the 20 amino acids that are available in nature. The only thing that is different between them is their side chain, which is shown collectively as R. And depending on what side chain there is, you get a different amino acid. Now, how do you make protein polymers? It's the same reaction that I've been talking about all the time. It's a dehydration synthesis reaction. A hydroxyl functional group is removed from the carboxyl end of an amino acid, and a hydrogen is removed from the amino end to make water, and then a covalent bond is formed between the carbon and the nitrogen, forming a peptide bond. So which part of this would be considered a peptide bond? This is it. That's the peptide bond. A peptide bond involves a carbonyl functional group and an amino functional group. Now, when you put amino acids together end over end, you kind of get like a long necklace. And these peptide bonds will attach these amino acids together end over end to give you a long polymer. But when you look at their structure, they can take all sorts of different shapes. These are examples of different proteins. So let's see if we can identify some of them. So this is a ribosome. That's the small subunit and a large subunit of the ribosome. You can see it's very globular. It has a particular shape. Then we're going to look at actin. This is a protein that is involved in contracting our muscles and other things. You see it has a very different shape than ribosome. Then we have alcohol dehydrogenase, which is an enzyme, very different from these two. And let's see what else we can. Lysozyme is an enzyme. Hemoglobin is a protein that is involved in carrying oxygen in our blood. So you see they are all taking different shapes. These are proteins that make the outer coat of a couple of viruses. This protein is an insect flight muscle protein. So what is it that why is it that we can start with protein molecules all starting as a chain of amino acids connect together end over end, but get all these different types of shapes? These different shapes depends on the type and sequence of amino acids that are included in the polypeptide. So when you look at this chain, when you add different types of amino acid and when you put them in different orders in the protein, it gives you a different shape. How are different amino acids different from one another? Well, depending on the class of amino acids that they're in, because of their R group, their side chain, they have different properties. And since they have different chemical properties, when they're placed into a polypeptide, they force the protein to take a particular shape. And the driving force of that is whether or not what a particular part of protein can interact with water or not, or whether it can interact with other parts of the protein. So let's take a look at these classes of amino acids. We have hydrophobic classes of amino acids. We have two different types of them. On the top is non-polar aliphatic amino acids. They have aliphatic side chains. By aliphatic means that they have carbons in, they have a long carbon chains. They're usually in a straight line. Aromatic nonpolar amino acids have side chains that have these rings on them. So anything that has these rings in them, chemists call them aromatic. But the big picture take-home message is that these amino acids, these two groups, they are hydrophobic. They are hydrophobic. They do not hydrogen bond with water or any polar or charged molecule through their side chains. Okay? So if a part of a protein has amino acids that are in these two groups, they usually turn away from the aqueous solution in which the protein is sitting, and it turns inwards. It hides from water. Then we go to the hydrophilic groups of amino acids. Why are they hydrophilic? Because they have either polar or charged side chains. So in this group we have polar side chains. In this group we have side chains that are positively charged or negatively charged. So when a part of a protein has these amino acids, these parts of the protein can easily interact with water, hydrogen bond with water, and these R groups also can interact with one another. So this allows the protein to take different shapes, and parts of the protein that have these types of side chains can stand out, can stick out and sit on the surface of the protein. Now, the sequence and type of amino acids in the protein is so important that sometimes a small change, sometimes just changing one amino acid in a protein, can have devastating consequences for the function of that protein and its structure. And a good example of that is within the hemoglobin. That's the protein that is present in our red blood cells that carries oxygen. Now, in normal hemoglobin, there is this amino acid sitting here with a side chain that is polar. And in water, actually, it's going to be negatively charged. So this amino acid is hydrophilic. The protein can interact with water in this part of the protein. Now, in sickle cell anemia, which is can be a deadly disease, this amino acid is changed, it's substituted because of a mutation in DNA to valine. And you can see that you're going from a hydrophilic side chain, an amino acid with a hydrophilic side chain, to an amino acid with a hydrophobic side chain. Now that part of the protein that normally could interact with an aqueous solution, all of a sudden it can't. So therefore, this causes hemoglobin molecules to come together and through hydrophobic interactions make long fibers, because really that protein can't exist as its free form in here any longer. It hides from water. So what's the consequence of these abnormal hemoglobin to form long strands? Well, first of all, they can't function properly to carry oxygen, and second, these change the shape of the blood cells, and these blood cells clog our arteries. They can't pass through the arteries anymore. So this can be a very deadly disease. So you can see the importance of having the right amino acids in the proper place in a protein for it to function properly. Now, how do proteins gain their structure? There are 4 levels of complexity in a protein structure. The first level is called the primary structure, and the primary structure is simply the sequence of the amino acids. It is simply the content, what amino acids are in the polypeptide and in which order they are in. Then we go to secondary protein structure, which is a little bit higher level of complexity, and this is when the protein folds in a local region within the protein to form either of 2 shapes, either an alpha helix, which looks like a helix, or a beta pleated sheet, which is kind of like accordion. This protein folds on itself. Then we go to the tertiary protein structure. That's when the protein, actually the entire polypeptide chain, folds on itself. And this usually forms what's called a globular structure, and usually most of the time this is the final shape of a polypeptide. Now sometimes, in order to perform a function, some polypeptides need to come together with other polypeptides to be able to deliver the function that they're supposed to do. So what they have to do is they have to come together with the other polypeptides. And when you have a structure, when you have more than one polypeptide interacting with one another, we say that we have a quaternary structure. That's the highest level of complexity in protein structure. Now, what derives formation of these structures? For the secondary protein structure, it is due to hydrogen bonding between the components of the polypeptide chain. We said, let me bring my pencil. So if we were going to look at a polypeptide if we were going to look at a peptide bond, this is a peptide bond, and also this is a peptide bond. So how does alpha helix or beta pleated sheet work? In this case, we're looking at a beta pleated sheet. We can see that there is hydrogen bonding between the nitrogen in peptide bond somewhere far away. So let's say this could be here, this one could be here, and at the bottom it could be this part of the protein. So there's hydrogen bonding between components of the peptide bonds. So in this case, in here would be the nitrogen, the hydrogen that is attached to the nitrogen. On this side would be the oxygen that is attached to carbon. Same thing happens in an alpha helix. So here is a peptide bond, and here's another peptide bond, and we can see that again the oxygen attached in the carbonyl part of this peptide bond is hydrogen bonding with the nitrogen, with the hydrogen that is attached to the nitrogen in a peptide bond somewhere close by in the protein. So the secondary protein structure is due to hydrogen bonding between components of the peptide bond. Write that down somewhere. How does tertiary structure form? Tertiary structure happens because of a number of different types of bonds. It could be hydrogen bonds, covalent bonds, it could be hydrophobic interaction, it could be ionic bonds, and the components of polypeptide that are involved in the formation of these bonds are the side chains. Okay? So interaction between the side chains of amino acids makes possible for the protein to fold on itself, makes it possible for the protein to fold on itself and take a particular shape. Now, you may have noticed that in this case, we have hydrogen bonds which are weak bonds, and in tertiary structure, some of them hydrophobic bonds are weak bonds. Hydrogen bonds are weak. Ionic bonds could be weak depending on conditions. The only type of strong bond is this disulfide linkage. This is a covalent bond between two amino acids that have a sulfhydryl side chain. And if you have curly hair, instead of straight hair, you have a lot of these disulfide linkages. And when you treat your hair to make it straight, you're basically breaking these covalent bonds. Now, the point I want to make with respect to the strength of these bonds is that because the shape of a protein is held together by these weak bonds, there's a lot of them, but still they're weak bonds, the shape of a protein is really susceptible to high temperature. And also because of a lot of these interactions in the tertiary structure and also quaternary structure has to do with ionic bonds and hydrogen bonds, the shape of protein also is very sensitive to extreme pH or extreme salt concentrations. For example, if you think the pH is really low, you have a lot of these positively charged hydrogen ions, which can come and compete with this entity for bonding with the negatively charged oxygen. So you have too many of these, you disrupt this interaction. Same thing could be true for the other end of the scale. If you have really low positively charged hydrogen ions, but then you have negatively charged, let's say, hydroxyl ions or any other negatively charged entity in the solution, then this can come and compete with oxygen for binding to this. And that's just an example. So, by review again, covalent bonds holds the amino acids together to form the primary structure. Hydrogen bonds within the same protein and between the components of the peptide bond are holding the secondary structure together. Hydrogen ionic and sometimes covalent bonding between R groups within the same protein are making tertiary structure possible. But what about quaternary structure? Quaternary structure, again, it's hydrogen ionic, sometimes covalent bonding between R groups of separate proteins. Now, since protein structure is sensitive to temperature and pH extremes, it's really important to have the protein in ideal conditions, ideal salt, pH, and temperature conditions. And what happens when we heat up a protein, just like when you cook eggs or beat up the eggs and you destroy the structure of the protein, so a lot of the protein in eggs is albumin. When you beat it up or when you cook it, this changes shape and basically you destroy the structure of the protein. And when we destroy the structure of protein, we call it denaturation. The protein denatures. You can visualize it in terms of these. If you have these paper clips, they have their nice shape, but when they're denatured, they lose their shape. And when they lose their shape, they change their function. Now, if you have a hard time visualizing these different protein structures, I keep on watching, and I have a demonstration of what it would look like. This is a demo I usually do in class, but since we're online, I created a demo of it as a video.